Welcome back, dear students, to video number 4 for Standard 7 Science. We are discussing chapter number 14. In the previous class, we discussed methods of separating mixtures. Let us continue and discuss a few in detail. So, we are going to discuss the first method, which is distillation. And for this, we need to take some salt water in a round bottom flask and arrange the apparatus as shown. So here we have a round bottom flask, there's a Bunsen's burner, there's a tripod stand, this is a wire gauze, and on which there's a round bottom flask, there's a thermometer, a stand which is holding this, and this particular tube is known as condenser. There are water inlets and outlets, and this is flask is known as conical flask. Inside this round bottom flask there is salt water. So let us begin. As I'm explaining, you please look at this particular figure or this particular apparatus. Start heating the liquid in the flask placed on the wire gauze. Slowly, droplets of water start falling in the conical flask. So when we start heating this water, after some time what happens is we see droplets of water falling over here. Now what exactly happens and why does water start coming over here? So the salt water in the round bottom flask boils on heating. So when the salt water boils, water in it vaporizes and it comes here. When the vapor passes through the inclined tube, this inclined tube, there is a condenser. It gets cooled because this, can you see this? Cool water circulates. See, there are two pipes. There's an inner pipe and there is an outer pipe. So cool water is circulating in this outer pipe, and the inner pipe there's water vapor. So because of cool water around it, the hot water vapor condenses and droplets of water are formed inside this. And since it is at an inclination, these droplets come over here and they start falling drop by drop into this particular flask. So now what do we have in this flask? Pure water. So salt remains here, water vaporizes up, condenses over here and falls down as pure water in this particular conical flask. So this method is known as distillation. Thus drops falling in the conical flask are of water from the salt solution in the round bottom flask. Salt remains behind at the bottom of the round bottom flask when all water has collected in the conical flask. This method of separation is called distillation. Distillation is also used for purification of impure liquids. So here we have shown how to separate water from salt water. It is also used in purification of other impure liquids. Next method. Method of separation using separating funnel. So first have a look at this particular picture shown. So what do we have over here? This particular funny looking thing is known as a separating funnel. There's a stand holding it. Here this is called a stopcock. You can open and close it. This is a beaker. And inside the separating funnel there's some liquid, there's kerosene and water. And now I'm explaining this. When I go to the procedure, you can have a look at this particular picture. So when a mixture of two immiscible liquids, what is the meaning of immiscible liquids? Liquids which do not mix. When a mixture of two immiscible liquids is left undisturbed, two layers are clearly seen to have formed. the heavier of the liquids remains below and the lighter liquids float on it. So we are using property of density. We are using a property of matter over here to separate mixtures. So two liquids in a mixture can be separated by making use of this property when their densities are different. So you can even separate oil and water. In this case, we are separating kerosene and water. So these are two immiscible liquids. They do not mix with each other. So what we do is we pour a mixture of kerosene and water in a separating funnel with the stopcock closed. So no liquid is allowed to flow from it. So this is closed, and we put mixture of salt and water over here. What happens is if we just keep this undisturbed, two layers are formed. Water is heavier, so it is at the bottom, and kerosene is on top. So after some time, what we do is fix the separating funnel firmly on a stand. Leave the mixture in the funnel undisturbed for a while. Water will remain below, kerosene will float on top. Now, without shaking the funnel, remove the stopper. So we open this particular stopper. Open the stopcock to collect the water. Now place a beaker underneath over here and collect the water till this kerosene comes here. When the kerosene comes here, quickly close the stopcock and you have water separated, and place another beaker. Open the stopcock, let the kerosene flow, and you have kerosene separated. So now kerosene and water are separated. So this is known as separating a method of separation using separating funnel. Let's go to next. Next is method of centrifugation. Now turbid water, ink, milk, buttermilk, blood are mixtures of liquids and insoluble solids. There are insoluble solids in this. These are liquid mixtures. When turbid water is kept undisturbed for a while, turbid means dirty water, the soil particles in it slowly settle to the bottom. The particles of milk or ink, however, do not settle, even on being left undisturbed. This is because the particles of solids in such mixtures, being very tiny and light, remain evenly distributed in the liquid. These particles cannot be separated from the liquid even by method of filtration or settling. So suppose we take a mixture of like milk. Milk is a mixture. There are milk solids and fat and liquid water and everything mixed in it. Or we take ink and we just keep those two containers, one containing milk and one containing ink, for some time like 2 hours and observe. Will you find solid milk or something settling? And can you separate water and solid particles of milk? No, you cannot do it. Same way with ink. Can you separate anything from that ink? No, you cannot separate by settling. Again, if you try to filter ink, will you be able to filter anything? Again, you cannot, because the particles, the solid particles which are mixed inside, are so tiny that they cannot be separated by filtering or by settling. So how will you separate such solid particles? In laboratory, there is a machine called as centrifuge machine. This is used to separate solids from a mixture of a liquid and solid. Now it looks something like this. It consists of a disc. You see this disc over here? This is an electrical centrifuge. So it consists of a disc which rotates like a fan. So it turns, it rotates like a, it rotates like a fan at a great speed. There is a provision to attach test tubes at the rim of this disc. So this is electrical, this is manual. You see this rotates and it has provision to attach test tubes. So this, it looks something like this. And when it starts moving, the test tubes turn and they become horizontal like this. But it is at such great speed that the liquid does not flow out. So exactly what happens when the tubes attached to the disc rotate at high speed. A force is generated which pushes the particles away from the center. As a result, the solid particles in the mixture in the test tubes collect at the bottom of the test tube and are thereby separated from the liquid. So, whatever is present, since this is moving at high speed, the solid particles are moved away and they collect at the bottom. You see this? And clear liquid is collected over here. This is how blood is also, plasma and red blood cells and white blood cells are separated in this manner as well. So this method of separation is known as method of centrifugation, and this machine is called as a centrifuge. Let's go to next. Next is method of chromatography. Now what is this? If two or more substances are dissolved in small proportions in the same solution, these substances are separated from each other by means of chromatography. This method is used in pharmaceutical science, factories, and scientific laboratories for detecting new ingredients and for identifying and separating components of a mixture. So let us see what exactly happens over here. So take some water in a beaker, take a rectangular piece of a filter paper. Put one spot of blue ink on it. So something like this, filter paper, put one spot of ink, blue ink, about 2 centimeters away from the edge, away from the bottom. Keep this paper upright. Now transfer this paper, just a minute, upright in a beaker. The beaker should have water, but the ink spot should be above water. See, paper is dipped in water, but the ink spot is not dipped in water. It is a little away from, above from water. So keep this paper upright in water in the beaker and place a lid on the beaker. After some time, the spot of the ink is seen to have risen. You will see the spot of ink rising up, going up, and fading up. If there are two or more constituents of different colors in the ink, they will be seen to have risen to different heights due to their different colors and thus appear distinct from each other. This experiment can be done using a chalk stick instead of filter paper. My dear students, you can do this at home as well. You will get filter papers in stationery shops. You can take those filter papers, cut into rectangular strips. On one, you can put a spot of ink, blue ink. On other one, you can put a spot of, you can use sketch pens also. You can put spots of sketch pens and you can place them in such containers and observe them. You will see various colored inks rising up like this. It is nice and interesting to do. In this method of separation called chromatography, two properties of substances are used. These are the solubility of the substance in the solvent that moves up. This is one property, and the ability of the substance to stick to the stationary filter paper. This is another property. These properties are mutually opposite and are different for different substances. As a result, all the components of the mixture do not rise all the way to the upper end of the filter paper but remain behind at different heights. So you will see different components at different heights due to these properties. So these were a few methods of separation. First one which we discussed was distillation. Then what we discussed was how to separate using a separating funnel. Then we discussed method of centrifugation, and lastly method of chromatography. So a quick recap about this chapter. Matter divided into one type of subunit called as pure substances and more than one type known as mixtures. Under pure substances, we have one type of atoms called as element, and we have more than one type, those are called as compounds. The smallest unit over here is molecule, and the smallest unit over here again is molecule but of same type of atoms. This molecule is of different type of atoms. Okay, these exist independently, they or are in combined form mostly, and these are all mostly always in combined form. You see, these are of same kind, these are of different kind. Okay, and here mixtures can be same or different. Ways of separating mixtures is using physical separation, and we discussed a few separation methods. Initially we saw pictures of sifting, winnowing, and all those various pictorial forms, and we discussed a few in detail like distillation, chromatography, centrifugation, and separating funnel. This is all in this chapter, my dear students. Please read the textbook, and notes will be given to you a little later. Write them neatly in your notebook. Have a nice day. Thank you for watching.